In this section of the Biochem Lab, we're going to be talking about protein purification. Protein purification is basically the art of separating what you want from what you don't want in the cell. If you are lucky enough to have your own research lab, you can choose an enzyme to work on for yourself. If you're working for somebody else, they're probably going to choose the enzyme that you're going to be assigned to work on. And then you have to think of things like choosing a source of the enzyme. So let's say you want catalase as the enzyme that you would want to work on. Well, catalase is found in high concentrations in peroxisomes, and peroxisomes are found in a high concentration in the liver. Now, you're not going to find too many humans that are willing to give up a piece of their liver for you to do research, but rats have very, very large livers. So if you're working on livers, rats are an excellent source of being able to get, specific proteins out of the liver. So this is how you would go about choosing a source of the enzyme. Then, when working with the enzyme, you want to choose conditions that are going to preserve the native state of your enzyme while you're working on it. You want to be able to preserve not only the activity, but also the yield and the purity of your enzyme. So there's some considerations that you have to think about when doing this. One of the considerations is which buffer are you going to use? There are quite a few buffers that you can choose. And initially, the buffers, and you'll see this slide again, so don't worry. Initially, the buffers that were available to biochemists were naturally occurring buffers, such as formate, acetate, succinate, citrate, phosphate, but they have very limited pH ranges. So you can see here's a pH scale, and you have these slides in your packet, so even if you can't see this clearly, please refer to your packet to see this picture. So you have a pH scale here. So if you are trying to work at a physiological pH, which is approximately pH 7.4, you can see that you're very limited in your pKa ranges of the buffers. Therefore, you're very limited in the pH ranges that you're able to use for these buffers. And if you're trying to work anything much over pH 8, you had almost no buffers that you could use at all. So this meant that someone needed to come up with a solution. Organic chemistry to the rescue. So Neil Good, an organic chemist, came up with what is called Good's buffers. He created synthetic buffers. So one of the problems that I have said with the naturally occurring buffers is that they had a very low pH range. Another problem with naturally occurring buffers is enzymes exist in the cell to degrade all of these. So coming up with synthetic buffers also meant that they were not biodegradable. And not being biodegradable meant that they could last for a long time in solutions. Now you still need to be aware of the structure of the buffers. You have a slide here of the structure of the buffers. You don't have to memorize these structures of the buffers. These are just some examples of Good's buffers. So you have things like bicene and caps and heaps and mops and tris and tricene. So all these different choices for buffers, each with slightly different organic structures to them. Now, one of the things you still have to worry about, even with synthetically created buffers, is that they are potentially having an inhibitory effect on any proteins. And you may not care, but it may be important to the overall functioning of what you're trying to do. And we will see this later in the aldolase experiment, where we have to very carefully choose the buffers that we are using, because some buffers may interfere with what we're trying to accomplish. But in general, if you have, for example, a tris buffer or a heaps buffer or something like that, they have some distinct advantages. One of the advantages is in a naturally occurring buffer, the temperature change can cause large swings in pH. But with these synthetically created buffers, the pKa does not change much with temperature, and if the pKa is not changing very much with temperature, the pH is also not changing very much with temperature. So very, very small changes in pH, so you can make it basically at room temperature and then put it in the cold room and use it in the cold room, for example, and not have to worry about huge swings in pH when you are trying to use it. So you don't have to have those structures and those memorized. That's just an example for you. Okay, so that is the idea of buffers. Now, the next thing that they would have to think about in protein purification is they have to decide what pH did they want to carry out their assay at. If you're doing alkaline phosphatase, your enzyme is going to have activity at pH 8. If you're doing acid phosphatase, your enzyme is going to have activity at pH 5. It may be that you have to try a bunch of different pHs to find, which pH works best. But my point is, a physiological pH of, say, 7.4 is not always the single best option for you. You also have to take into account some other factors for the enzyme and maybe use a range of pHs to see which pH is going to work best. You have to consider ionic strength when working with proteins. Too high of a salt concentration, and if you have some loosely associated factors that are binding together to make a wholly active protein, too high of a salt concentration, and maybe you can dissociate those factors that are coming together to make an active protein, which will result in an inactive protein. On the other hand, too little salt, and you can get proteins aggregating together and precipitating out of solution. So there will be some ionic strength that will work best for your protein. You have to consider what temperature you are isolating the enzyme at, or that you are doing your assays at. If you have a temperature of 55 degrees Celsius and you're doing an assay at 55 degrees Celsius, your half-life for an average enzyme might be 2 minutes. But some enzymes, like Taq polymerase, will work fine at 55 degrees, will work fine at 72, even can withstand 95 degrees. If you're doing an assay, for example, with Taq polymerase at 37, it's not going to be as active at 37 as it is at 72 degrees. But other polymerases or other proteins that you're trying to work with, you get it too hot and it completely shuts down completely. If you're working with some types of polymerases and you even heat it up to 45 degrees. I was using a reverse transcriptase one time and was doing a temperature gradient on there, and when you get much past 42, 43 degrees, it was shutting down and having very little activity. On the other hand, my husband was working on an ATPase, and he was isolating an ATPase, and his lab could not figure out why their ATPase did not have good activity when they were finished purifying it. And so went to another lab to see what they were doing. They had horrible lab techniques. They weren't keeping anything on ice. They didn't keep anything cold, but they had good published results with their ATPase. But that turned out to be the key, because when you chilled that particular enzyme, it actually dissociated at cold temperatures. So that's one thing you have to think about, is that there's no one temperature that works all the time for every enzyme. You might need to try a couple different things when you're setting up an assay to look at your protein activity. So sometimes metal ion concentration, a particular metal ion concentration, is required. Some enzymes might need a certain magnesium concentration or zinc. If you don't have any zinc at all in your cell, in your solutions, you have some proteins that will have no functionality. So some would require calcium, for example. So you can have different metal concentrations that are required by enzymes sometimes. You have to consider protease activity. And when we're isolating proteins, we try to do whatever we can to minimize the protease activity. And proteases can come from the fingers. Proteases are present in the cell. There are particular things that you can do. You can keep it generally in an average assay. You're keeping everything cold to minimize protease activity. You're wearing gloves so that you're not introducing more proteases into the solution. So you can have specific protease inhibitors that you add as an extra to your buffers, for example. So you're trying to do things to minimize protease activity. Sometimes you can have enzymes that might be sensitive to light. Not very many, but it happens occasionally that you have an enzyme that is sensitive to light, and so you might need to do things a little more in a darkened environment to preserve your activity. So when you then are working on things, you need to consider how you're going to break open the cell. So you've worked on an assay for your particular protein, and now you need to break open the cell. So there's different ways of breaking open cells, and these can be used in conjunction with one another. It's not only a single way. So the first way is freeze-thaw, and in a freeze-thaw procedure, you are freezing it, usually like in liquid nitrogen or something, very, very cold, not just like on ice, but really freezing to a very low temperature, and then quickly thaw it, 37 degrees to thaw it quickly. You do this several times, and this is putting microfractures into the membranes. This may be enough for your particular cell that you're working with to break open the cellular membrane and have the contents released into whatever buffer you're using. It's not always enough just doing a freeze-thaw procedure. Sometimes these are going to be used in conjunction with one another. Another thing that you can do, very often in conjunction with freeze-thaw, is sonication. And in sonication, you are using energy to make a very high-pitched 20 kilocycles of sound, and because this is able to break open cells, you need to wear earplugs when you are doing this, because it can also break open the cells that are in your ears, those little hairs that are in your inner ear that help you hear. It can fracture those if you spend too much time near a sonicator. So that's some of the safety issues you want to think about if you are doing sonication. So basically, sonication is taking energy and it's funneling this energy down to a little tip, and that tip vibrates at 20 kilocycles, and the sound waves that it's introducing into your cells, which are in solution, are breaking open your cells. Now, this much energy is also generating heat, so you want to make sure that you have your samples on ice when you are trying to do this, or you're just going to simply be heating up your samples and denaturing all the proteins that you're trying to isolate because the heat is causing denaturation of your proteins. So you want to be somewhat careful with that. Another method is using differences in pressure. This was originally called a French press. Now there's an instrument called a microfluidizer, and the idea behind the French press or the microfluidizer is that you have sample contained in an environment, and you're putting a lot of pressure on that sample. In the original French press, it was manual pressure. You had like a piston that you were pushing down on, very mechanical. In the microfluidizer, it's using air pressure to increase the pressure on your sample. In any event, whatever method you're using, whatever type of instrument you're using, you are really increasing the pressure on your sample very, very high, and then you are opening the spigot and letting your sample come out. And this change in pressure from high pressure, where it's acclimated to a high pressure, to boom, all of a sudden being allowed out into a lower pressure, tends to burst open a certain fraction of your cells. And then you put it back in there, and you do this several times in rotation so that you get a significant number of your cells broken open from these differences in pressure. Another method that you can use is autolysis. So basically, if you add toluene to your system, like if you have yeast or something like that, you can add toluene to it, and this triggers some particular lytic enzymes to start lysing the cell membrane, the cell walls from inside. Basically, it's hydrolyzing itself. This isn't used very much at all, but still might be used with specific cell types occasionally. Osmotic shock. In this set of experiments we have coming up, we're going to be using osmotic shock. And osmotic shock is basically using these different salt concentrations. So if you have a cell membrane or a cell wall, and you have a very, very low solute concentration outside of the cell membrane, and a higher solute concentration inside the cell, which is generally the case because you have lots of salts and things like that inside the cell. So you have a cell that's bathed basically in distilled water, and the pores that are in every membrane open up to let the water inside the cell. The membrane is trying to equalize the solute concentration inside and outside the membrane. So it allows water inside the cell trying to equalize it out. You're not letting the solutes out into the solution, but you are letting water into the cell. So you're letting more and more water into the cell, more and more water into the cell, until finally, boop, the cell bursts open. So that is osmotic shock, that difference between a high salt concentration inside and a low salt concentration outside of a membrane, allowing transfer of water which can burst the cell. You can use particular enzymes. So if you have E. coli, for example, and you need to degrade the cell wall for E. coli, there's something called lysozyme. You add lysozyme to the cell wall of E. coli, and it will start chewing away the peptidoglycan layer on the cell wall of the E. coli, basically make the membrane accessible to the outside fluid, and then you can use some detergents to be able to lyse open the cell once the membrane is available. Now, sometimes you might need to use more rudimentary methods. Grinding, if you have plant material, grinding is a method that's used all the time. So you might have a leaf, for example, maybe a very waxy leaf, and you're trying to get some of those cells out of that waxy coating of the leaf. So you take a mortar and pestle, and you're just grinding that with the mortar and pestle. Sometimes people add sand into it to give a layer of helping break open the cells. And so the grinding in that mortar and pestle, that grinding action, can help break open all the cells and release the proteins. Sometimes when you are finished with one of these earlier fractionation procedures, you might use a homogenizer, or you might even use a homogenizer in the beginning to break open your cells. So some of the different types of homogenizers. You have a Potter homogenizer, and in this case, a homogenizer is able to work because there's a very, very low clearance between a Teflon-coated pestle inside and a glass container outside. There's very, very little clearance there. And so if you put a solution down here and then press down, usually with a drill press or something like that, so a lot of force down here, you are forcing the solution right here by the side of that Teflon-coated pestle, and there's very, very little clearance. There's enough clearance there just for sometimes a single cell to pass by. Some have clearances that are so small that even a whole cell cannot pass by. So until you have maybe a smaller organelle that has been freed from the cell wall, you don't have enough clearance for it to pass by. So there's homogenizers with different apparent clearances to allow different sizes to pass by the pestle as it moves up and down. So basically, you're putting pressure on the solution is being forced to to pass by this small clearance, and it's only allowing a certain size of material to pass by. And then of course, after you finish pushing it down, then you want to pull it back up, and it pulls it back on the other side. So you can do this several times, and by the time you've done this, back and forth, maybe 10 to 15 times, all of your cellular material has been broken open, has been reduced in size. A Downs homogenizer is basically the same idea, but instead of having a Teflon pestle in the middle, usually it has a glass pestle, so it's glass on glass. Glass inside, glass outside, and it tends to have smaller clearances. So for example, when I was working in a biochem lab, I would use a Downs homogenizer to be able to isolate nuclei from whole cells. The first thing I did is I isolated whole cells, then I used a particular Downs homogenizer to be able to separate out just the nuclei from the whole cells, because it had a different level of clearance. Now, if you have very tough material, you can use a polytron homogenizer. And a polytron homogenizer uses sort of sharp blades that are pointing down, and there's one set of blades to the outside and another set of blades to the inside, and one set will circulate around the other set. So one set stationary and the other one will circulate. The inner one will usually circulate. Very, very sharp. I managed to cut my arm one time on a polytron, and all I was doing was I was reaching across the countertop, and I forgot how sharp that blades in the polytron were, and it scraped along my arm, and it opened up a nice big cut on my arm just from barely touching it. So very, very sharp blades. But if you're working with something like a chicken gizzard, for example, which is very, very tough, very muscular tissue, something like that would be necessary to be able to chew up all that high amount of connective tissue and be able to free the cells out of that. An Omni-mixer, if you've ever just had like a little milkshake mixer at the store, an Omni-mixer just sort of has a blade to mix everything up. On the other hand, a Waring blender, we're going to be using a Waring blender in our first photosystems experiment. And a Waring blender, like the kind you make drinks out of or smoothies, just uses a blade to chop open the cells. So basically, there's some way of getting all the cells open. So after you've gotten all the cells open, then you have to start saying, well, how am I going to assay this particular protein? So you have to have some way of doing an assay of your protein, measuring how much protein you have. So there's different types of assays. And again, you have this page, and you're not going to be able to read this up here if you're watching on video, but you have this page in your packet. So there are a number of assays up here. So if you have acetylcholinesterase, for example, acetylcholinesterase as an enzyme takes acetylcholine and it's producing acetic acid. So you can titrate the amount of acetic acid to be able to say, okay, here's the activity of my particular enzyme. Some others, if you have a polymerase, for example, polymerase is one of the proteins when I was up at UC Davis we worked on, and so we would have radioactive nucleotides, radioactive DNTPs, and so the amount of radioactivity that was incorporated into the DNA that it was making, that gave us a rate of reaction of the enzyme. Or it can also chew it up. So if you have a deoxyribonuclease, which is chewing up DNA, you're going to get an increase in absorbance at 260 because all these individual DNTPs are being freed from the length of DNA. So there's all these different kinds of assays. So you can have a chemical assay, you can have a spectrophotometric assay, you can have radioactive assays, all different types of assays, just depending on the activity of your protein. You don't need to memorize these are just some examples. Okay, so for identification and tracking of protein, as I've said, you can have chemical or spectrophotometric assays, you can have radioactive assays, you can have immunological assays. So if you have a particular antigen and you're trying to measure the amount of antigen, you can mix it with an antibody and you can do something something called an electromobility shift assay, gel shift assay, and say how much, does this when the antigen binds the antibody, how much movement is there, because now it's a larger size, so it's going to move through a gel at a different rate when they're together versus when they're individual. So you can have immunological assays. You can have reactions with ligand. You can have receptor ligand, glycoprotein, lectin, so different types of assays to be able to track your protein. But when you are talking about assays, a practical assay needs to be possible to perform the assay rapidly on many samples. So if it takes 2 days to do an assay and get the results from the assay, your protein is degrading while you are waiting. So you need to have an assay that's going to be able to be run fairly quickly. It needs to reliably indicate the amount of the desired protein present at various stages of purification. So let's say you break open your cells and you want to be able to separate the cytoplasmic contents from the whole cells and membrane parts and stuff like that. So you spin it in a centrifuge. Well, if in your centrifuge, if you're trying to do an assay and say, is my protein in the supernatant? Is my protein in the pellet? If you don't have an assay that can reliably say how much protein is present in the supernatant versus the pellet, you're not going to know. If all your assay can say is, yes, you have some protein in the supernatant, yes, you have some protein in the pellet, you don't know whether to save the supernatant or save the pellet. So it has to be able to indicate the amount of the protein that's present in the various stages of purification. A good assay should not significantly deplete the overall yield of the enzyme from the preparation. So if it takes 50% of the enzyme amount to be able to get a good assay on it, well, you can purify a bunch of protein, and let's say you do a supernatant versus pellet, and it takes 50% of your protein just to be able to tell that. Well, then the next step, it's going to take another 50%, and the next step, it's going to take another 50%. And maybe by the time you're finished with a couple steps of purification, you have very, very little of your protein left just from the assays that you've had to do on that. But just because you have less and less protein at every stage is not necessarily a bad thing, because remember, protein purification is basically getting rid of the protein you don't want while keeping the protein that you do want. But losses are going to invariably occur. If you have 10 fractionation steps to purify an enzyme, and each step had 80% efficiency, at the end you have less than 20% of the starting material. But that might be okay with you. There is a procedure that we were doing in a lab I was working in where we were working with Drosophila embryonic proteins, and it's a very, very low copy number of Drosophila embryonic proteins. So we would start off with several hundred grams of Drosophila embryos, and by the time we were finished with the 8-step purification protocol, we had so little protein that you could not even do certain assays on it because we didn't have enough protein left over. We could know that our protein was there, but there's so little of it, but it was pure. And that was the important thing, that it was pure and that we needed very, very little of it to be able to continue on to be able to study our protein. So it depends, if we'd had 20% of our starting material, we would have been like, woohoo, that's great. We had even less than that. So it just sort of depends on what you need versus what you have. So when you are doing this, doing your assays, you want to be able to determine how much protein you have. And that's great. If you have enough protein that you can dry it down, that's fantastic. And then you can just say, measure it out. I need 10 milligrams of protein, great. Just take some of my protein that I have dried down, get it under a balance, measure out 10 milligrams, hey, great. A lot of times you're not going to have even 10 milligrams of protein if you're working with a very, very rare protein, for example. Maybe you have it in solution and you have just, very, very little of it. So you don't necessarily have enough to get a dry weight of your protein. But again, if you have lots of it, you can use some of these intrinsic properties to get a refractive index. If you have a lot of protein, hospitals use turbidometric methods to measure total protein. For example, if they suspect you have kidney problems, so that you're having protein leak through your kidneys and giving a high protein concentration in your urine. So what they will do is they will add a chemical which precipitates all the proteins out and turns it turbid, and then they can measure the turbidity of the solution as a way of saying what's the total protein in here. If you have enough, great. You can do AA analysis on it if you need to. But on a practical level, most of the time you need to use more subtle methods rather than just being able to get a dry weight, for example, on it. So, other ways of measuring how much protein you have while you are going through a purification process, there are absorptiometric, non-destructive ways to measure your protein. Now, non-destructive is important because maybe it takes a lot of solution to be able to measure it, but if it does not destroy your protein, you still have it to put into the next step, for example, that you're looking at. So, you can measure at an absorbance of 215. And at 215, you're measuring your actual peptide bonds. But the downside of using 215 to measure the absorbance of proteins is you have buffers that are interfering with that and also absorb at that same wavelength. A lot of buffers also absorb at that same wavelength. But the good thing about using 215 is because it's measuring the actual peptide bonds, remember the carbon-carbon nitrogen backbone of amino acids, it's actually measuring those. It's independent of the side chain residues, so it doesn't matter, whether you have tyrosine, tryptophan, glycine, how much you have of each. It's independent of those. Another absorptiometric non-destructive method is reading the A280. So reading the A280, it's measuring basically aromatic amino acid residues. Now, this means you have something called a reference protein problem. And a reference protein problem basically says the more you have of a particular amino acid, the higher the absorbance you're going to have. Now, if you're measuring just some very specific amino acids, if you have, for example, in the protein you're measuring, a greater amount of tyrosine, well, you're going to have, if you have one protein that has twice as much tyrosine as another protein, you're going to have a higher A280 just because you have more tyrosine in there. Not because you actually have a greater microgram per mil or a greater concentration of your protein, but just because you have a higher level of the particular amino acid that's being measured by the technique that you've chosen. So this is called a reference protein problem. Let me give you another example. So this is a particular assay. It's a Bradford assay, which we're going to talk about in a minute. But if you have two different proteins, so this is BSA and this is IgG, and you can have an identical concentration, so you can have 0.5 mg per mil of BSA, or you can have 0.5 mg per mil of IgG. So they're the exact same concentration. But if you are doing an assay on them that is reacting with certain specific side chains, the BSA is going to have an apparently higher concentration than the IgG does, just because the assay that you've chosen reacts more with those certain side chains that BSA tends to have more of than IgG does. So, for example, if you are using A280 as an assay, and if a protein that you are working with has a greater number of tyrosine amino acids, then it's going to have an apparently higher concentration than another protein, which might be the exact same concentration, but has fewer of those residues that are absorbing at A280. But 215, because it's actually measuring peptide bonds and it's independent of side chain residue that's being measured, the more protein, more total protein you have, the higher the A215 you're going to get. But remember, you have a lot of things that can interfere with reading it at 215. So generally what we do to acknowledge there's that reference protein problem, if you are using a method to measure your protein that does have a reference protein problem, we say things like, with reference to BSA, here's my concentration, or with reference to IgG, here's my concentration. So you're not saying, here's my absolute concentration that I know for sure. You're saying, comparing it to a solution of the exact same, of a particular concentration, comparing mine to a known concentration of IgG, or comparing it to a known concentration of BSA. Here is my apparent concentration of my protein. So, in addition to these absorptiometric non-destructive methods, you have some colorimetric, although destructive methods of determining the concentration of proteins. So the first one I want to introduce you to is a Biuret assay. And a Biuret assay basically measures how much nitrogen is in your protein. So it's using copper to react with nitrogen, and when the copper reacts with the backbone, the carbon-carbon-nitrogen backbone of the amino acid, and the copper reacts with that, it's causing a particular absorption at 550 nanometers. So the greater the absorbance you have at 550 nanometers, the more protein you have. Now something like this, because it's not reacting with the amino acid side chains, with the Biuret assay, there is no reference protein problem. Why doesn't everybody just use a Biuret assay? Well, it has other problems that we're going to be talking about in just a minute. There is no one perfect assay. There's just the best assay for the conditions that you have chosen. So, in addition to the Biuret assay, there's a nitration assay. You may have done a nitration on yourself. If you've ever worked with nitric acid, for example, your fingertips may have turned brown. And when your fingertips turn brown from working with nitric acid, that is nitration. So it works with very specific amino acids, and in the presence of those amino acids, it turns brown, and it can be picked up at a wavelength that will say, okay, the more brown the solution is, the higher the concentration of those particular amino acids. Now, the higher concentration of those amino acids generally goes with more protein, but if you have a protein that you're measuring that's higher in those particular amino acids compared with other proteins, again, you can have a reference protein problem because you are specifically measuring amino acids and not saying we're measuring the carbon-carbon-nitrogen backbone of the actual peptides itself. Likewise, the Sakaguchi reaction uses arginine, and in that one, it's using naphthol and hypochlorite. So Sakaguchi reaction measures the naphthol and the hypochlorite and gets a pink color. So the pinker the color is, or the more absorbance at that particular wavelength, the higher protein concentration you have. So in addition to the Sakaguchi reaction, you can have a BCA reaction. And the BCA reaction is similar to Biuret. So first, in a BCA reaction, first you do a Biuret assay where you're reacting protein with the copper, and it makes copper with one positive charge, and then that copper then reacts with 2 molecules of BCA, which causes a complex, which then can be picked up at a specific wavelength. And because the initial reaction of the copper is not reacting with the side chain, it is instead reacting with the carbon-carbon-nitrogen backbone and not dependent on any of the amino acid side chains, BCA, again, does not have a reference protein problem. So the next one that we can talk about is the Lowry assay. So in the Lowry assay, you have phosphomolybdotungstate. And phosphomolybdotungstate specifically oxidizes tyrosine. So in this case, in the Lowry assay, you have tyrosine and you have the phosphomolybdotungstate, which originally is yellow. After it reacts in an oxidation-reduction reaction with the tyrosine, it turns blue. And so the bluer the color, the more tyrosine you have. But if you happen to have a protein which has more tyrosine side chains than another one, you're going to potentially get an inaccurate number. You can have, like generally higher, but with reference to a particular protein, here is my concentration. So it has that reference protein problem for the Lowry assay. Another method is the Bradford assay, and the Bradford assay is what we're going to be using in this class when we have to measure how much protein we have. So this is Kumasi Brilliant Blue, and this is the backbone of the Bradford method. So again, you have particular side chains which are reacting. The dye is binding your hydrophobic amino acid residues, basically. And after it binds those particular residues, the maximal absorbance of the dye changes from 465 nanometers to 595 nanometers. So the greater the absorbance at 595, the greater the amount of the hydrophobic specific residues that you have that are reacting. So again, has a reference protein problem. So there's a nice little table here that's, and I realize you probably can't read this, but again, it's in your packet of information. And so this is the kind of thing that you would need to know for an exam, basically. So here's the different most important, most used methods, because not very many people do a Sakaguchi reaction, not many people do nitration to determine the concentration of protein, but people still use Biuret and Lowry and Bradford, BCA, and spectrophotometric methods. So these are the most commonly used ones. Now, I said that if you have something like a Biuret assay, that there is no reference protein problem. So why doesn't everybody just use a Biuret assay all the time to determine the concentration of their protein? Well, it has very, very low sensitivity. So it can take up to 20 milligrams of protein to be able to get an accurate number of a concentration, and it takes quite a bit of time. It takes about 30 minutes to do the assay. And there's the principle here, the basic principle that I told you about, that you have the peptide bonds reacting with copper and it turns it purple. There are some interferences, so if you're using one of Good's buffers, or if you have tris in there and some other particular amino acids, those can interfere with the reaction. On the other hand, the Lowry assay has much higher sensitivity. You can get a good, accurate protein concentration with as little as 5 micrograms of protein, but it's a much slower reaction. It can take up to an hour to do. And again, here's the principle, which I just talked about, of why you're doing it, and here are some interferences. So if you have precipitated your protein with ammonium sulfate, and some of that is still with your protein, you can't use it because that will interfere with the Lowry assay. It has very critical timing for the procedure, so you need to make sure that you're getting your timing down. If you let it go for more than 60 minutes, you're going to have different apparent concentrations. The Bradford assay, this is the one that we're going to be using, has a very, very good high sensitivity. You only need about 1 microgram of protein, and it's fairly fast. You can do it within 15 minutes, sometimes even less if you're very, very efficient. And tells here the principle that that maximum absorbance shift when it's reacting with the particular amino acid side chains. There are some interferences, Trimex 100, STS, you're using detergents or strongly basic buffers. You could not use a Bradford assay. So that's why you can't just use a Bradford assay, say, all the time. The BCA also has very, very high sensitivity, but it is very slow and it has some different interferences. So this is what you should be knowing for the exam. And this is our non-destructive method, the spectrophotometric method. You need about 50 to 100 micrograms of protein to be able to get a good reading off of it, compared with, say, 1 microgram for a Bradford assay. You need 100 times more protein, but remember, it's non-destructive, so that you still have the protein afterwards. You have not destroyed it, you still have it to use. It's very, very quick, and it works by the tyrosine and tryptophan residues absorbing at A280. So this is some of the methodology that we can use. Teresa, stop the video.